Okay, good morning. So as Arthur says, we're now handing over from the more exciting future-focused moonshot policy aspect of things through to the kind of hardcore technical nuts and bolts for a little bit. So I hope everybody has had some coffee. And I figured it was due a content warning that for at least the next little bit, there will not be pretty pictures, maybe the occasional pretty picture. There will be schema diagrams, at least occasionally, and there will even be the odd JSON document. And on top of that, there will be acronyms, and I've already used one. So JSON stands for JavaScript Serialized Object Notation, by the way. Sorry about that. So I'm going to try and give an overview, and I'm actually giving the first talk in this session, but this bit is more background as to what the purpose of the morning's theme focus on ontologies and metadata is about, with reference to what we're trying to do technically in the project. And I'll talk more about that in a moment. But what we wanted to do first was, because we're going to be focused on these topics for the next few hours, including the breakout sessions, and then in the afternoon, the panel debate, is talk about what we actually mean by ontology and metadata models. So quick check, who has heard of ontologies? That's a great start. Who would be happy to come up here and define ontology in front of everyone else? Okay, oh, that's more than I might have expected. That's great. Okay, I'm not going to ask you to do it because that's not necessarily fair. I'm going to attempt to do it myself, but I'll probably get it wrong, so feel free to correct me. So idea here is to establish some shared terminology, and I do that in 3 bits. So it's useful to think about splitting this task of understanding how to describe the metadata component of images into 3 bits. And the first bit is the data model. So data model is kind of talking about trying to define what things we are talking about. So in the case of images, we want to talk about images, but when we start to talk about images, we probably also want to think about what the images are of. There's some kind of sample. There's maybe some kind of preparation for that. There was an imaging instrument that was used to capture the image. But then when we get into reporting about these things, we often need to think about context. So why was the image created? What were people doing? Who were the people? So we start to put these diagrams together, which basically relate the entities involved. As soon as you do this in the real world, it tends to get more complicated. So I polled somebody in one of the talks over the last couple of days, mentioned the ISA, Investigation Study Assay Model, which is used in quite a lot of different bits of life sciences to describe data. And from ISA's website, I pulled the full chart of their data model. And it shows that, again, they sort of got 3 principal things, which are the ones in red: investigation, study, and assay. But as soon as you start to describe real world data from that, you need to add things. So they've added publications and organizations, people, but also materials and measurements and things. And there's a sort of slightly optimistically over to the right ontology reference, which doesn't quite link to anything, which I thought was quite funny because it shows that they use ontologies, but doesn't describe exactly how. And then a note of caution. And the reason this is a little bit hard to see is because this is a picture of a capture that one of my colleagues was giving at the retirement event for another colleague, Alvis Brasma, who was really involved in partly in setting up the BioImage archive. But this was going back 20, 25 years to the birth of another data resource called ArrayExpress for gene expression data, when hybridization and arrays were the really exciting thing, and that was the data that everybody wanted to share. And they showed how they started with this sort of simple block diagram of all the things you need to describe arrays, hybridizations, experiments, and samples. And this looks kind of reasonably simple and comprehensible. You have to think about it a little bit. And then again, when they got into implementation, the result looked more like this. And this is the sort of warning that these are the processes that we are going through collectively at the moment. We can put up simple diagrams about data models, but as soon as you get into the weeds, it gets more complicated. Okay, so data model is what are the kind of things we're talking about. Next part, and sometimes these are merged, and I think it's useful to separate them for a reason I'll talk about in a moment. For each of these things we talk about, you then want to start to understand what's useful to describe about that thing. So for example, for a biological sample, you maybe need to think about describing it. You need to think about, perhaps what might have been done to it in terms of experimental treatments. You need to think about what organism it came from. There were probably more things than this list. You need to link it to other things. So you go from model objects to things you need to know about those model objects. So data model is, again, is kind of what things we're talking about, metadata schema. What do we need to know about those things? Final part, and we heard quite a lot about this over the last few days, is how we're going to describe those things. And I really like in Stephen Jett's talk the other day, the looking at trying to compare slides, histopathology slides, and thinking about the different ways people have described what we instinctively recognize as the same concept. So if you've got a slide and you've got lots of labels which are trying to convey what we immediately recognize as humans, as being these all are talking about, say, thoracic cancer, these are all different from a computer's point of view. These are all different strings. If all we have for this thing is the exact words and we're trying to compare the exact words, these are 5 different things. And that's a problem for machine interoperability. And a problem for machine interoperability is a problem for training AI models. And that's a problem that we nowadays end up caring about a lot. I mean, we've always cared about it, but big driver now is AI. How do we get around this problem? This is where ontologies start to come in. So there is a way to solve this problem, and the way to do that, or one of the ways to do that at least, is to use an ontology. And this is an ontology called the human disease ontology. And there's kind of lots of information in this slide, but one of the things to focus on here is basically a unique identifier, a kind of world resolvable identifier that uniquely describes thoracic cancer. So, great, maybe we can use this. Like data models, this becomes more complicated as soon as you start to look at it in detail. One way to illustrate this is down the bottom right here. And what this means is basically, okay, there's actually cross-references. And the reason for the cross-references, there is not one ontology for everything in the universe, kind of bore hairs aside. There are different ways because there are different domains that are trying to use ontologies, and they often overlap. And one way to help solve that problem is to look at cross-references. So for example, there's cross-references for the concept of thoracic cancer in another ontology called SNOMED. And if you go and look at SNOMED, you can look at an identifier. So this is another unique identifier, another way to solve the problem for the same concept. Same concept, but sort of, because when we go back here, if you look very carefully, which you probably can't do because it's probably too small, but there are 2 entries here. And the reason there are 2 entries here is because there's one for malignant neoplasm of thorax, and there's one for just neoplasm. And those are things that matter a lot in certain circumstances for diagnosis. And kind of the problem here, and when we start to look at ontologies, is that they're describing concepts, and concepts don't always overlap. So sometimes people will say, okay, it doesn't matter which ontology we can choose because we'll just map between them. We'll take one term in one and map it to another term in another. You can't always do that. It's like translating human languages. Words have meaning that is not always mappable to another meaning in a different language. So ontologies kind of help with this, but they also create other problems. The people that put their hand up when asked if they define ontology would probably at this point tell me, I'm actually not really defining an ontology. I'm defining a controlled vocabulary. This is a list of terms. So another reason why we need an ontology, because an ontology is really terms and relationships, I think is nicely illustrated by that. So it's quite reasonable to come to an image repository and say something like, okay, what light microscopy images do you have? And this turns out to be a harder question to answer than you might think, because we have, for example, bright field imaging, where we've recorded an imaging method which is bright field. We have light sheet imaging, where we've recorded light sheet, or maybe spin. We have super resolution imaging, where we might have recorded the imaging method as D-storm. We have histopathology imaging, where again we might have recorded it as bright field, maybe with some caveat about being histology. Again, we'd all recognize these as being light microscopy. How do we relate them? And that's where one of the other properties of ontologies come in, is that they have relationships. So in this case, lots of information here, but we're looking at a biological methods, a bioimaging methods ontology. And this has a kind of tree structure. And down here, what we're seeing is that there are different forms of light microscopy. And so we can look at, for example, describing SPIM, so light sheet, sort of, as being one type of fluorescence microscopy, which is one type of light microscopy. So using the ontology there helps us to be able to answer these questions about what are, say, light microscopy images in the database, because we can look for things in this tree which eventually have a parent that is light microscopy. So ontologies encode both things and relationships. And again, anybody who's really pedantic might say, okay, it's a taxonomy. Ontologies can have more complex relationships. I won't cover that just for the moment. Last thing I'll say, PIDs. Joy gave a great presentation on the importance and use of PIDs a couple of days ago on Wednesday. PIDs are very useful because they uniquely describe kind of an instance of a thing. So an ontology might describe the concept of a person, but we know that there are lots of different people. It's useful to uniquely identify those people because you want to be able to track research outputs of people. Now, from our side, it's really nice that I can have my publication history and dataset outputs associated uniquely with me, and my name is not that uncommon. There are lots of people with the same name, so it's hard to resolve me just through my name. My organization has changed over the years and will change in future, so that will change. ORCID provides a unique reference. This is my ORCID identifier that is kind of always me. And that's useful for tracking within and across domains. The talk on Wednesday, I think from Maria from DataSite, was talking about the research organizations register, and that's a way to uniquely identify organizations. So this is a unique identifier for EMBL EBI. So these tools together help us solve, or these are the things that go into putting together metadata schema and vocabularies. These are the tools we have to work with, but it's useful to think about them separately. And one of the reasons I think it's useful to think about them separately, and there will be discussions in the breakout room and for many years down the line about harmonizing these things, is because in general, it's easier to change things, particularly for existing databases on the right-hand side. None of these things are really easy to change when you have existing data, which is why the more agreement you can do in advance, the better. But on the other hand, it's hard to get everything right, so you do need to update. Changing the unique vocabularies you use is possible. I mentioned some of the challenges about mapping, but there are potentially ways around these. Changing, for example, data models in existing databases is a huge, huge amount of work. It's much, much, much more difficult. You sort of have to rewrite everything. So you need all of these things together, but it tends to be easier to align and change the right-hand sides, harder, but possible for the middle sides. Left-hand side is much more difficult. And also, you know, if you change the way you're describing these concepts, you then have to usually change the things further to the right. Because if you change, for example, you know, you split sample into two different types of objects, you will then have to go back to the metadata schema and work out how you split those and maybe update those. So these are kind of the tools and concepts to think about going into this morning session, some of the breakout sessions, and then the panel.